Welcome to the Quality Assurance element, Common Element B. This module will focus on quality assurance requirements and considerations that are common to all field monitoring activities that involve measurements or sample collection. The first topic introduces some basic concepts about data of known quality and shows a matrix of activities that need to be done to affect, check, record, and report the quality of data. The second topic provides the big picture of the quality assurance process and emphasizes the importance of the actions taken in the field. Assuming that you intend to generate data of known quality, your role is to assure that the data will be delivered to the data users with information about two major aspects. First, how good the data is in terms of accuracy and precision of the measurements or analyses. And second, what the data represents in the environment. This element, Common Element B, takes care of the first aspect. Common Element C, later on in this course, discusses the second aspect, including the importance of tailoring the where and the when to your specific monitoring intent. You already know that when you conduct measurements and collect samples in the environment, you encounter both inherent variability and measurement error. Sources of inherent variability abound. They are results of natural processes, and they cannot be prevented or reduced. Sources of error can and should be controlled, and you will learn how to implement various procedures for that purpose. The following screens of this element show you how to handle the measurement and sampling error. Once you can handle that, Common Element C will show you how to distinguish between inherent variability and measurement and sampling error. Click the button if you want to go there now. You've heard lots of folks talk about quality assurance and quality control, but you're probably not always clear about what they mean. The original definition of quality control included a loop of three processes: specification, production, and inspection. It worked really well for production lines of interchangeable machine parts. When that term was applied to quality of scientific measurements, another term was added: quality assurance. Today, there are many definitions for quality assurance and quality control. What you see on the screen are the definition used by SWAMP, and they are very general. Quality assurance is a system of principles to assure data quality, while quality control refers to detailed activities. The definitions received from SWAMP Region 7 are also helpful. Quality control is defined as a single step or procedure that is performed to evaluate a single aspect of the analysis or test. And quality assurance is defined as the sum of all the quality controls performed in the laboratory plus everything else that is done to produce reliable data. This field methods course is about what you, the field operator, actually have to do to produce reliable data of known quality. So it translates these definitions into the actions that need to be done to affect, check, record, and report the quality of data. And yes, avoidance of the words assurance and control is intentional, and so is the color coding for these actions. Quality assurance is about several distinct aspects of data quality, including accuracy and precision of field measurements, sample integrity, process reproducibility, and the operator's proficiency. You can assure and communicate each of these aspects through the following types of actions: affect, or act to influence outcome; check, or test to evaluate or verify; record, or keep everything documented; report, that is, communicate the data quality indicator and the overall quality of your data. This matrix shows you what you can actually do to affect, check, record, and report the quality of your field measurement data and the integrity of your samples. Let's look at accuracy as an example. Accuracy is how close a measurement is to the true value. It is instrument specific, and it refers to all field measurements collected with a given instrument during a given period of time, usually between calibrations or accuracy checks. To affect the accuracy of your adjustable reading instrument, you simply need to bring it to the calibrated state often. You should also clean the instrument, kit, or test tube before and after each use, and protect all field equipment from extreme temperature, sunlight, excessive humidity, and harmful liquids or vapors. This will help ensure accurate function of the instrument. To check the accuracy, Conduct accuracy checks every time before calibration adjustment. Be sure to use certified standards for all calibration and accuracy checks. To record your accuracy, capture the instrument reading before calibration and the true value of the standard. To report accuracy, calculate the difference between the instrument reading before calibration and the true value of the standard and express it in the measurement units, for example, 0.5 degrees Celsius, or as a percentage of the standard if appropriate. Precision is how close our measurements are to each other when we take repeated measurements at the same time and the same place. It is also used as a measure of the reproducibility of the entire process of sampling and analysis. As you walk through this matrix, you will see that you need repeated measurements to assess precision in the repeatability sense of the word, and you have to take field duplicates when collecting samples to assess precision in the reproducibility sense of the word. Sample integrity refers to lack of contamination, which you can assure by keeping everything clean, and check by way of blank samples, and lack of deterioration, which calls for chilling the samples immediately or preserving them when taken, and for temperature blanks to be checked upon arrival in the lab. Blanks are an essential part of checking and reporting data quality, and there are several types of blanks as you will see later in this element. Going through the different rows of this matrix will help you familiarize yourself with what you, the field operator, need to do to affect, check, record, and report the quality of your data. By the way, COC stands for the chain of custody process you have seen in Module 6, and RPD stands for relative percent difference, which is the difference between two repeated measurements expressed as a percentage of their average. Please refer to the SWAMP toolbox for more information on error calculation. The list is not that long if you boil it down, and not that confusing if you separate it by each action. To affect the data quality, calibrate your instrument frequently, clean it after each use before it dries, and be consistent in the way you do things. To check how good your data actually are, conduct accuracy checks and repeated measurements, and collect blanks for analysis. The functions and use scenarios of the blanks you may need to collect when you are sampling water are described in detail in the next screen and in Section 4.6 of Module 4 in this course. To record everything, make sure that your data sheets have all the essential placeholders. See Common Element D for more information about this. To report, you or someone else down the data quality management line of operators will need to calculate the accuracy and precision errors for inclusion with the data in the database. As you recall, sample integrity is a very important aspect of data quality, and it is your responsibility to prevent contamination and deterioration when you conduct field activities. It is also your responsibility to check sample integrity, or at least initiate the checking process by collecting blanks. The EPA provides the following definition for the word blank in the context of field work: a sample that is intended to contain none of the analytes of interest. A blank is used to detect contamination during sample handling, preparation, and/or analysis. You probably know that there are several potential pathways of contamination. Blanks address these pathways separately and differ from each other in what they intend to check. The initial equipment blank is a sample of clean water that has been run through the pre-cleaned equipment prior to shipping the equipment out into the field. The trip blank, also known as the container blank or travel blank, is a sample container of the same batch used for the environmental samples that has been filled with clean water. This blank is shipped and analyzed along with the other samples to check for, or confirm the lack of, container or shipping contamination. A field blank is a sample of clean water that is shipped into the field and run through the same process and equipment as the field samples. For grab sampling, the bottle may only be opened during the sampling. A temperature blank is a container filled with clean water that is shipped together with the field samples. When the lab receives the samples, the sample custodian measures the temperature of the water in that bottle and records this on the chain of custody form or in the logbook. The functions, frequencies, and use scenarios of the blanks you may need to collect when you are sampling water should be described in detail in your quality assurance project plan. There's not much you can do in the way of blanks when collecting sediments. However, proper decontamination is essential, and water rinsette blanks are necessary for the sediment sampling and compositing devices you have used. As you have already noticed, Actions to assure data quality are associated with most of the field activities described in the 6 modules of this course. To put things in perspective, the chart on your screen shows you the big picture of the major steps in the quality assurance quality control process, including data verification and validation. You can see how the rest of the quality assurance process leans on the work that you, the field operator, do to satisfy the requirements shown in the top 4 boxes. Whether you are conducting measurements, collecting samples, or documenting information that describes the data, what you do provides a sound basis for data of known quality. However, please note that this scheme relates only to the "how good are the data" question, not to the question, "what do the data represent in the environment?" Roll over the boxes in the top part of the flowchart to familiarize yourself with the actions you will perform and the tools you can use. Making sure of all quality assurance actions to affect, check, record, and report data quality takes many steps, but these steps are done in a logical sequence and can be handled easily by one person for a small monitoring effort. However, if you have a large monitoring effort, with multiple operators doing different tasks related to the same monitoring activity at different times, you need a lot of coordination and communication, and you also need good planning. For example, Joe and Dale come back from the field with retrieved data loggers at 7 p.m. Stephanie and Mark do the post-deployment accuracy checks and calibration adjustments later that night, while Kim changes the dissolved oxygen membrane and Nellie prepares the map and station finders for the next deployment. Then, Dale calibrates the DO electrode in the morning before picking up all the data loggers and going out with Ann to deploy them in the new stations. And this is just one example. The logistics of quality assurance is finite, but it takes organizing, and having clear steps, tasks, roles, and responsible personnel is helpful.